My name is Nkechi Emea Ibisi from Michael Ofbara University of Agriculture, Omodike, in Abia State, Nigeria. I'm glad to be here today and to be given this opportunity to make this presentation of my work. The topic of my presentation is adsorption and corrosion-inhibiting effect of Aspilia africana extract on low-carbon steel corrosion in acidic media. Case study. Today, all over the world, most of the things we use for our structural materials are low-carbon steel. And the reason is because Really? Okay. The reason is because of the strength. 2 is malleable and also is ductile. And also is easily available because the cost is very low. But if you check around, most of the things we use are low carbon steel. A majority of these materials on-the-go corrosion. What do we mean by corrosion? The simple definition that we can always use sometimes is rust. And that's a layman's interpretation of what corrosion is all about. Now, why did I put up this? If you go globally today, a lot of money is being spent on corrosion. And there's a record today that about billions of naira are spent annually in trying to handle the structural materials that we use for so many things, and namely in our pipelines, industries, engineering, in your homes. This thing is everywhere. So, and we spend money on maintenance. We spend money on even building the structures and all of that. So there is a lot of money that is getting into corrosion. So, and we've been like, what will we do? Because if globally we are spending so much money on that, what can we do? Because a lot of economic resources is going into that, and that can be used for other things like capacity building and a lot of other things. So we're looking at that's a big time problem. It's not only for you, it's not for me, it's for everybody. So we are saying, what do we do about that? So there are certain things that we're looking at, and the insight you have here is just because we've had several solutions. If I say that a lot of money is going into that, that means that all around people are working at that to see that corrosion is being stopped. And there are materials that are being used to inhibit corrosion or to stop corrosion or to make sure that corrosion does not continue. But we are looking at this because we are trying to look at an electronic structure pattern to be able to say, if there are things that we can use, are there ways we can harness it better to make sure that we'll have better results? That's why you have that insert. And as I get along in the work, you get to understand that better because that's part of the work. Now, the objective of this study. This present study has a dual purpose. One, first of all, we want to establish further the effectiveness of plant extracts as corrosion inhibitors. Plant extracts are being used today as corrosion inhibitors. And before I say that, there are a lot of work today that have been done on corrosion, but organic materials and inorganic materials are used for corrosion inhibitors, used as corrosion inhibitors. But recently, they had a lot of attention on organic materials, and not just organic materials, but plant materials, basically. And why did we do that? We've been talking about eco-friendly materials, things that will not hamper our environment and all of that. So there was a lot of attention on that. And still, basically, if we still have all of that and a lot of work have been done, so many plants are being used, why do we still have more work being done? And that's the question, and that question can be answered through this work. And we also want to attempt to deduct the inhibition mechanism that will be, and the possible adsorption modes of the extract's active components. Now, You can never talk about inhibition without talking about adsorption. Why? The materials that we use, because in corrosion you have an adsorbent and you have the surface that is going to be adsorbed on. So there's no way it can happen without that surface, so there's got to be adsorption taking place. Now, in recent times, density functional theory, that's a based quantum chemical computation, have proven to be very useful in elucidating the electronic structures and reactivity of a wide range of organic molecules. And it has become common practice to incorporate quantum chemical calculations in organic corrosion studies. And here we have used, and I've applied the DFT techniques to assess the adsorption behavior of this material we have worked on. And remember that what we have worked on is a plant material, and we made an extract of it. Now, we have an outline which incorporates the introduction, experimentals, and the result and discussion and all of that. I have spent some time talking about the corrosion, what it is, and all of that. But for a chemist will not just look at corrosion just the way every other person look at it. Even though we say it's rust, but we want to get down to what is happening. And a chemist look at corrosion as an electrochemical process being taking place. And why is that? Because there's transfer of ions taking place. And in that state, for any electrochemical process to take place, there must be a cathode, there must be an anode. There must be one accepting electrons, there must be one donating electrons. And what happens during corrosion is that there's transfer of ions, basically. So that's what we're talking about here. And you know, our electrons, the electrons that are involved, usually act as ions as well. So when we talk about those electrons, that's why we say we are the study, the DFT, that is an electronic study process, always deals with transfer of electrons. I have two methods here that we have used, the gravimetric method and the theoretical modeling simulation. In the gravimetric method, basically is about weight loss. And most of us know what weight loss is. The simple way to calculate what is happening, if a rust or if an ion is exposed to an environment. Now, I need to go back a little to tell you that there's no way rust can take place without oxygen, and because there must be environment involved, oxygen and water. So it's basic. So if that's going to be happening, and if I'm talking to a layman, I always say, this is a metal, and for this particular study, we are talking about a carbon steel. And another name for it is mild steel. And if that mild steel is existing, and it comes in contact with an environment, and that reaction begins to take place, which means we have an ion, we have oxygen, we have water. It's actually expressed in the first structure that I gave you here. And when you have a combination of that, this is what you have. You have the ion at the anode, we have the oxygen and the water at the cathode, and the combination of all of that, this is what you have. And you know, this is what we call rust. That's what we call rust. Now, for a layman, immediately an ion begins to absorb that, and reaction begins to take place. It begins to lose weight. That's why we talk about weight loss. And the simplest way for a scientist to look at that, you now check what is happening. Can I actually measure what I had put in and what is happening after the process? Is the weight before and after the same? So we use it to check the weight and to find out. That's why we use the gravimetric method that measures the weight loss, to find out if actually there's something happening. And another parameter that's so important, it happens and it is involved in every environment that corrosion takes place, is the temperature. Temperature is usually involved. So we also had to conduct the study using different temperatures to find out what happens when the temperature is high, when the temperature is low, and for whatever type of temperature there is in a particular environment. So what is it that happens? Now, as a useful method to protect metals and alloys deployed in service for aggressive environments, usually what we call aggressive environments, like the seawater, usually is an aggressive environment. If you have an acidic environment, it's an aggressive environment. For this study, is an acidic environment because we had used, in our study, we had used 1 molar HCl. So it's an acidic environment that we're talking about here. So it's an aggressive environment. So you find out that the corrosion, if you add species into the solution in contact with the surface, in order, you can actually inhibit the corrosion. And when we talk about the species now, it's like when I introduce the inhibitor, and for this particular study, we are using as a plant material, and the extract of that plant material is a species that can be introduced. It reduces the corrosion rate and also affects the corrosion reaction. Now, coming to the material, we use parts of the material. In a study like this, because from literature we have ascertained that most of the most plants that are used for medicinal purposes have been proven to be active as corrosion inhibitors. And there are certain things that are common amongst all of them. Most of them have phytochemicals as composition, and those phytochemicals have things that are species that are important, like polar atoms. You find things like nitrogen, sulfur, and oxygen, and all of that. So they are things that help to make them very potent as corrosion inhibitors. So, and before we make a choice of material that we are going to use, and that's a choice of a plant, we must find out whether that plant is available. That's if it is easily accessible to. We also find out would there be, because there are also plants that could have harm or cause harm. So we will find out what kind of plant are we using, well, is it easily available, and all of that. So if we're able to identify that, then we'll make a choice of a plant. This particular plant was gotten and within the resources in the Michael Opera University of Agriculture, and the masteel that we used were from Federal University of Technology, Owerri, and they were treated before they were used. Coming to the results now. Now, I want to also step a little back and talk about what we do when we are using density functional process to that's computational processes to also look at the electronic structure of a material. If we have a material like this one, and after we have confirmed that we can work with it, I didn't do the isolation for this particular one, but because the isolation had already been done. So there are a lot of phytochemicals and also materials and that have already been isolated from this. So what we did is that we just took one out of them, and for this particular study of interest, to find out if after modeling that structure And are there things that are involved, like are there elements, are there composition, the way the composition of this material, will it be able to be an effective inhibitor? So we first of all did the modeling, and after the modeling, we now did what we call optimization. For any particular compound to be stable, it has to be optimized. And we used a computational package that is called the mole. So the mole is a very good density functional theory package that we can use. So we used it. And there are certain parameters that are chosen to be able to use this, this package. So I had inferences, so I chose the parameters that will be specifically used for this, and after choosing that, we did, after optimization, based on the optimized structure, I can now use the optimized structure to now look at other parameters. We looked at total electron density, we looked at the orbitals. The orbitals comprises of the highest occupied molecular orbitals and the lowest unoccupied molecular orbitals. So we looked at those parameters. Those are the things that form the electronic structure. So for this particular study, we limited ourselves to those basic studies. We also took care of the effect of temperature. Like I had already said, the inhibition efficiency was also worked on. I think I should use this now.
There you go.
All right. I had talked about the weight loss. We had two basic samples of the plant material. We had the stem and the leaf, and this shows the weight loss versus the concentration. We use different concentrations to check the ability of the stem extract and the leaf extract, the effect. This is the efficiency against the concentration of the two samples as well. We also have this to show the concentration of the leaf extract. Now, there's another study that we made. We also try to find out if there's a synergistic effect between the inhibitor and we use an additive. The additive is potassium iodide. If you add an additive usually to the inhibitor, the efficiency it increases. How does it? For particular extract, so you have to work on it to be able to have that inference. So we used a concentration, a basic concentration, and with the potassium iodide to check if the inhibitor, because this shows, this is the blank, and this is also the blank of the potassium iodide. This is the inhibitor alone. That's the weight loss. This is concentration with the weight loss, and this is the weight loss when we had the inhibitor and the potassium iodide. So with the synergistic effect, the weight loss was quite low. Yeah. So we shows that there was an effect of the additive. The same thing that we had. These are two different samples for the stem and for the leaf. This is about the effect of inhibition efficiency against the concentration. Now, this is efficiency against temperature. Now, like I also said, we use isotherm. usually there's no way you can do an adsorption study. The isotherm helps you to actually study the adsorptions on the that is done on the surface. So we use that to relate and we did 3 different ones: the Tenkem isotherm, Friedrich isotherm, and Langmuir isotherm. Now, here comes this is the optimized structure of inositol. Like I told you, this is already an isolated compound from this particular plant. So we just took it and modeled it and used it for this study. So this is the optimized study. Before we can ever begin to look at all of this electronic structure, this structure must be optimized. And it's from this optimized structure that we can now use, because when it is optimized, it is stable. So at stable, when we say stable, it has the minimal energy. So we now use it to do these studies to check the electron density. These 2 are orbital studies. One is called highest occupied molecular orbital and the lowest occupied molecular orbital. 1 minute? 2. Okay. All right. Okay, as I round up, the electronic properties of the inositol, I've already said this. From there, you can see the blue and the yellow isosurfaces depict the electron density difference. Then the blue region show electron accumulation, while the yellow region show electron loss. There are colors here. So the tendency of a molecule to donate to the vacant d orbitals with ion atom is reflected by high values of electron highest occupied molecular orbital. So we usually find the difference between this and this to be able to know, and this will help us to have a better interpretation of the structure and what is happening. So from this study, right after we do the computations, the computer gives you the values, and we are able to write those values and find that I didn't put the data here. All right. Okay, based on the results, the following were summarized. Since we have this problem, we found out that Aspilia africana is a good green inhibitor for mild steel, which is also, I told you, it can be interchanged, mild steel or low carbon steel. And increase in extract concentration increases the corrosion rate. The corrosion of mild steel in HCl increases with increase in concentration of the acid and also with the period of contact and temperature. Like we talked about, if the mild steel is exposed to the environment, with the timing, that's why we keep changing, because some of this we do for a certain number of hours. We check it within 72 hours or especially this one was done within 72 hours. So you keep checking. If you put an example, you have to put it because the contact time is important. Now, immediately, no matter the level of contact, immediately the metal surface touches oxygen or no matter what it is, it must not be immersed in an environment like being in seawater and all of that. Corrosion must take place. All right, okay. Lastly, corrosion of Aspilia africana is attributed to chemizoption of the chemical phytochemical components of the extract on the surface of the material. Well, acknowledgements. This work was partly supported by University of Science and Technology, China. These are the references. We used their supercomputers, and it was combined with the Shanghai supercomputers also. They usually have them inside the university, so we were able to use that. Thank you very much.